Muted.
Program Director at the University of Texas MD Anderson Cancer Center in Houston, and it is my pleasure to welcome you to AGT's webinar series. For those of you who are not AGT members, please consider joining the organization so that AGT can contribute to be the voice of technologists in the laboratory community and be able to offer programs like this in the future. Please check AGT's website at www.agt-info.org to see the many benefits of our memberships. I joined AGT 20 years ago, and over the years, I have established many friendships and has provided me with many opportunities both professionally and personal growth. A few housekeeping notes before we get to today's presentations. Please feel free to ask questions during the session. In the upper right side of your computer screen, you will see a control panel. In the lower portion of that panel, you can type in a question or comment and submit it to the webinar organizers. You can do this at any time during the presentation. We will reserve about 10 minutes at the end of the presentation for question and answer. If you are participating as a group, please designate one person in the group to complete the online evaluation. In order for everyone to receive their certificates of attendance, that person will need to enter the names and email addresses of each person who is participating at your site today into the evaluation. As a backup measure, a sign-in form can be completed and faxed to AGT as well. Certificates will be emailed to participants approximately 30 days following the webinar. Today, Dr. Newsham will address next-generation sequencing as a clinical tool, platforms, and possibilities. Dr. Newsham received her undergraduate degree in biochemistry from the University of Vermont, followed by a PhD in applied biological sciences from MIT, following postdoctoral training at the Ludwig Institute in Montreal, Canada. She has held faculty appointments at UCSD in San Diego, Virginia Commonwealth University, and University of Texas MD Anderson Cancer Center, where her laboratory focused on cancer genetics and applications technologies for oncogenomic analysis. After leaving MD Anderson in 2007, she served as a genetic analyst consultant for Beckman Coulter, and in 2009 joined the Human Genome Sequencing Center at Baylor as a senior manager for the library and exome capture groups. In 2013, Dr. Newsham rejoined MD Anderson Cancer Center as an assistant professor, education coordinator for the Molecular Genetic Technology program in the School of Health Professions. In her current position, she educates and trains undergraduates in molecular genetic technology who will become board certified to work in the field of clinical molecular diagnostics. And now I will turn it over to Dr. Newsham.
Thanks, Peter. Good afternoon, everybody. Thank you for taking your work and lunch break to attend this webinar. So I was asked to sort of give a primer on next generation sequencing and discuss some of the issues as it relates to this becoming a clinical tool. So on the introduction slide, I've just given you one of my favorite timeline cartoons, starting from 1865 when Mendel discovered the laws of genetics. And actually what's not on there is 1875 when Friedrich Meissner first identified DNA. But you can see how many years it took to get to the Human Genome Project, and then how much happened between 1990 and 2003. And of course now we're a decade after that. But the technology that was developed during the Human Genome Project has actually revolutionized molecular biology and also is now revolutionizing clinical healthcare. So what I'm going to try to do, if it'll let me switch my slides, here we go, is just at first give background on the different platforms that are next generation sequencing platforms, and I'm going to focus more on the two which are fairly standard now in the molecular diagnostics labs. That would be the PGM or Ion Torrent and then Illumina, but also give some homage to the original other platforms which are still around. And then I'm going to talk a little bit about whole exome sequencing because that was the standard use until now. The cost of next-gen sequencing in terms of clinical is come way down. And then I'm going to just go over what myself and some of the leaders at the Human Genome Centers feel are the challenges for adapting next-gen sequencing as a molecular diagnostic tool. So on the slide I'm showing now, on the left, as you can see, we're all familiar with Sanger sequencing, where you would fragment the DNA, or you would actually have a single piece of DNA that you would clone into a plasmid, isolate individual plasmids, and then traditionally use dideoxycycle sequencing to generate different fragments. So just briefly, these are nucleotides that have fluorescent tags. They're not able to incorporate a next nucleotide because they're missing the hydroxyl group. And so by doing a series of, in the PCR machine, a series of cycle sequencing reactions, you generate a ladder of fragments whose fluorescent tag identifies the nucleotide that was last added. And so typically these are run on capillary instruments. ABI 3500 is the one that our students work on. And you develop an electropherogram, which is color-based, that you can translate into the sequence. So these are the Sanger sequencing methodologies that have been around for decades now, and they are still sort of the gold standard in terms of validation of mutations that are found by other means. But during the Human Genome Project, other technologies were developed to facilitate the rapid sequencing that was needed to generate the entire genome sequence. So we still start out with DNA fragmentation, but in this case, we add on adapters on the ends. And then there are several different methods that are used to sort of expand these fragments to the point where you can have a visual sequence. They're called colonies or clusters, and I'll go over that in the next few slides. But in different ways, these different technologies give you a way to image what nucleotide has been added, color-based, and in that way you can generate your sequence. So the obvious advantages of next-generation sequencing is that you're sequencing a library, so you're not sequencing an individual piece. And you don't have to do any in vivo cloning or transformation or colony picking, which, if you've been around as long as I have, you've enjoyed so much of that. And the other thing is it's array-based sequencing. So it's a much higher degree of parallelism than a capillary-based sequence. Those capillary instruments are, they can only sequence as many as you have capillaries for. So in this case, you're generating multiple colonies that you can actually get a good sequence for and generate whole exomes and whole genomes from that. Okay, so for next generation sequencing, you really can break it down into 3 individual steps. There's the library preparation, then the clonal amplification of those fragments that you've generated, and then depending on the platform, the cyclic array sequencing. And right now, the libraries generally, they have different adapters, but the actual methodology is pretty much the same between the different platforms. Where they start to distinguish themselves is how you generate those amplified sequences or clusters. So in several cases, you use an emulsion PCR, and I'm going to go over this in more detail in the next couple of slides. And those platforms were originally Roche's 454, which was based on pyrosequencing, the solid platform, which was based on sequencing by ligation, and now in the last 15 years or so, sequencing by pH, which is the personal genome machine or the ion torrent. So those machines all use an emulsion PCR to generate the clusters of fragments to sequence. The other methodology for generating clusters is called bridge PCR, and this is associated with what was originally Solexa, but we now all know as Illumina, and that technology uses sequencing by synthesis. So all of these are high speed and high throughput. You're able to get qualitative and quantitative sequence, and what took 10, 13 years you can now be done in as little as 10 days on the newest technology. So I'm going to start out first, as I said, by focusing on the two platforms which are in the highest use in the clinic. And first I'm going to talk a little bit about the Ion Torrent. This is actually an instrument that our students have access to, and we spend the last 3 weeks of their senior semester generating Ion Torrent libraries and running them and doing data analysis. I think this is an excellent educational tool for them to really get a feel for what's going on in next-gen sequencing. And then I'm going to touch on the Illumina platform as well. So just briefly, there's a picture of enrichment, which are two smaller instruments. Of course, now they have an all-in-one plug-and-play system, which some of the diagnostic labs are bringing in. But for the Ion Torrent, it's a very simple methodology. There are many steps. As I told my students, there's about 40 steps in the library preparation. But basically, you're shearing your DNA to a particular size, which is typically 150 to 200 base pairs. And you're adding the ions specific primers to allow you to amplify. And once you've amplified those DNA fragments, you partially digest the primer sequences down, and you use the individual adapters that work for the ion sequencing. So you can, and this is true for all platforms, you can add the traditional adapters, or you can add adapters that have a barcode. And those barcodes are 6 to 8 base pair random sequences, which will identify your particular DNA from a particular sample. And if you barcode, then you can actually combine multiple libraries into one sequencing run, because the bioinformatics out the other end will bin your sequencing results based on what barcode sequence they have. So depending on the chip that you use to run this on, you can multiplex up to 30 different libraries in a single run. So that's very useful if you're looking at throughput. So you're generating your DNA fragment with your two particular adapters on that help you do the emulsion PCR and the enrichment. Okay? So it's not necessarily rocket science. It's just very particular, and it's very detailed. Once you generate your library, as I said, you have to amplify. So you have to form clusters to get enough fragments for you to generate a readable sequence. And so in the case of 454 and Solid and now the Ion Torrent, they use emulsion PCR. And this is a picture from Roche's website about their emulsion PCR. This one is from the Ion Torrent, but they both say exactly the same thing. You have what's called here ion sphere particles, and they have a sequence that binds the P1 adapter of the fragments that you've generated. And if you mix your PCR reagents and your DNA and your spheres in a particular ratio of oil to water, you will generate droplets. And those droplets are calculated to include one sphere, one DNA fragment, and enough for you to amplify. And so through that process in the emulsion instrument, you are generating a sphere that starts with one, but that ends up with many copies of the same DNA fragment that has annealed to the P1 adapter that's on the sphere. So this is what they call the cluster formation. So you have many copies of one DNA fragment piece on one bead. Okay? And the same is true for 454. That's a very powerful technique in order to amplify your fragments. And the ability to generate individual PCR machines, thousands of them in a single test tube, is what allows you to generate thousands, tens of thousands of these spheres that have specific DNA fragments that you can then sequence on the Ion Torrent. So we're not talking one fragment anymore, we're talking in the tens of thousands. Okay. So for the Ion Torrent, it's actually a chip-based design, and it's as I tell my students, it's I like to call it zillion, but it's many pH meters on a chip. And so you will load your clusters through a port onto this field. And this field has a layer of wells, that fit one bead each. Okay? And then it has a sensory layer and an ion sensor, which is effectively your PCR or your pH meter. And so when you load your chip, you dump one sphere with the cluster of DNA into each well. And then one at a time, you will push a fluid with one nucleotide. So you have to do each nucleotide separately in this case. And as you pass, for example, an ATP over the top of this well, if there's a T which will add that A, okay, as it's shown over here, it will release the hydrogen, and you will be able to detect that as a change in the pH of the solution in that well. So it's just each sequencing reaction is really an individual pH reaction each time that you pass a nucleotide over the top of the well. And so you're doing this in thousands of different wells at one time. And that signal is then sent to the ion torrent server, which generates your results as not only qualitatively, but quantitatively, that if you're adding a C here, that's what you're adding. And so the beauty of this, in one sense, is there's no fluorescence. So in terms of cost, it's cheaper. And the fact that this chip design is based on factories that are out there that know how to make these chips, you're not really having to develop any high throughput means or technology to manufacture a lot of these. They can use a lot of the technology that was worked out for computers and other things. So when you think of the ion torrent, just think of it as a multiplexed pH meter. And each time you pass a nucleotide over, if it finds its complement, it will release a hydrogen ion, and you will detect that as a change in the pH. So just to pay homage, as I said, to the originals, the Roche 454 was one of the first instruments out there for next-gen sequencing. And the Genome Center, when I joined some years ago, had several of these because the error rate was so low. It was a very hardy instrument. It took a long time to run, but the data you got out the other end was a very high quality. But as you can see from the slide, it's the same technology in the sense that you have a picotiter plate where you're dropping these individual beads where you've formed your clusters. And then when you're adding your nucleotide, it will release a pyrophosphate. So this is based on pyrosequencing. So your beads are added, you add your incubation mix, which has your polymerase, and then you add this bead, which has the two enzymes that are needed to do the pyrosequencing and also the light detection. And when this pyrophosphate is released, when you add a nucleotide, you will generate an ATP, which works with the luciferase, and it generates a light signal. And so you have individual light signals appearing right on your plate here. So if an individual well has added a nucleotide, it should generate a light signal, and then that is translated by the software into sequence. And if you have two A's in a row, the light signal should be twice that. So again, it's qualitative and also quantitative at the same time. The problem with the 454 in the end was that it's very slow. And so obviously it's all about how fast you can do it and at what quality. The other next-gen sequencing platform that I was going to give you a slide for, which I think is, no, I don't know where it went. It's missing, sorry. I was going to tell you about the solid system. So when I was at the Genome Center some years ago, they had an entire fleet of the solid platform. And they used that for a very long time, again, because the quality of the reads coming out were excellent compared to other systems. So this sequencing by ligation, you have your DNA that you've pretty much prepared the same way with adapters, and you've made your clonal amplified clusters. And you have 16 combinations of oligos that have at the end, and whatever combinations of two nucleotides, so AT, AC, AG, etc. And you pass those over, and wherever that oligo binds, right, it will generate a fluorescent signal for you. And so that's why it's called sequencing by ligation. It's actually quite a cumbersome technology. It takes a very long time. It used to take, I think, more than a week to run a single sequencing run. But the data it gives you out the other end is good because you're moving one nucleotide each time, but you're interrogating two on the end of your oligo. So each base is kind of sequenced twice. And so that's a sort of validation in the sequencing platform itself. So if I run across the slide, which I had earlier, I'll stop and explain it again. But it's a cumbersome technique and very slow, but because you end up interrogating each nucleotide twice, it tends to give you a very low error rate. But as most of these two platforms suffered from, what usually puts things in the background is that they're not fast enough. So they're not generating enough data in a short enough period of time. So the other technology is based on this bridge PCR, and it's Illumina, and it's called sequencing by synthesis. So I'm just going to go back over the library construction so you can see that even for the Illumina, the flow for most of the library construction is pretty much the same. You fragment your DNA either enzymatically or with ultrasound to a particular fragment size, which again is about 150 to 200 base pairs. You size select in that range. In this case, you repair the ends, and then you add an A on the end. You adenylate it, and then you bring in your Illumina-specific adapters, and you PCR amplify using these adapters. So again, you're starting out with a series of fragments of a particular size. You're just adding on Illumina-specific adapters, and then you're amplifying those to get enough information to do your clonal amplification. And so for this, the insert size could be between 200 and 500 base pairs. And as always, there's always Q&A in the flow to make sure that your libraries are of good quality. And then if you're going to be multiplexing them, you have to normalize them to the same sort of concentration. I wanted to point out that Solid and also Illumina had another library construction that was often done, and this was called mate pair library. So as you can see, you start out with genomic DNA, you shear it, but to a larger size. You put the adapters on, and then they have a protocol where you actually form a circular fragment. So you're actually connecting the two ends from your fragments by circularizing. You're connecting the 5' end of this, for example, 2 to 5 kb piece with the 3' end. Okay? And that's what's happening here. So here's your adapters and this coupler that you put in here. And then you get rid of the DNA in between, because really what you want are the ends. Okay? And you again circularize, and you put a junction code in there so you know where the two ends came together. And then through another exonuclease digestion, you create this linear fragment, which can now undergo clonal amplification by bridge PCR. But what you've done is you've actually jumped a sequence that's 2 to 5 kb. So you'll sequence here and you'll sequence here, and this will help you put scaffolds together or longer pieces of DNA without having to sequence all the way through the 2,000 to 5,000 base pairs. Okay. So this is just a short slide to show you can do single read on the short, 200 base pair libraries you make. Each Solid and Illumina developed paired ends, so you could sequence from the 5' end and the 3' end, so effectively sort of doubling the sequence that you could get. And then in terms of the mate pair, you would be sequencing both ends so that you could scaffold a genome together without having to actually sequence all the thousands of base pairs in the middle of that piece. And this is just a list of, the paired ends are much better for detecting rearrangements, useful when more data is required, Mate pairs were especially useful when you're doing new genome assembly and resolving regions that have complex repetitive elements. Okay, so this is a diagram of the cluster or bridge amplification that Illumina uses. So they have which is on the next slide, they have something they call a flow cell, and that has 8 lanes in it. And those lanes are coded with these different sequences that are homologous to the 2 adapters that you use to generate your individual fragments. Okay. And you flow your fragments over the top of these, and where they find their complementary sequences, they will bind. And then you actually run a PCR. And what that does is allows you to amplify specific fragments in a specific area. Okay? And they will bind to each of them and then create a double strand, and then you do a denaturing, and then it will do it again. Okay? And this is all done on the flow cell that you'll actually run in the instrument. And using this bridge amplification, you can actually generate millions of molecular clusters. So this will represent a sequence, right? This will represent a different fragment sequence, and so on. Okay. So this is, whoops, hang on. This is to give you an idea. So you've generated your fragments. You have your 2 different adapters. So A1 might be the red, A2 would be the blue. These are the sequencing primers. They're actually different on both ends, so you can actually sequence from both ends in a paired end fashion. And by doing this bridge amplification, which is the equivalent of the emulsion PCR, you generate batches or colonies of the individual fragments, which then you can now run the sequencing reagents over to sequence. Okay, so this is sort of a diagram of the flow cell, right? You put your fragments in here. It's coded with these 2 different adapters that will bind your DNA fragments, and then through running the PCR reagents over it, you will generate individual clusters right here in each of the lanes. And as I said, the surface is coated with the primers, but then during the sequencing run, you just flow the reagents in one end and out the other, right? And after each cycle you wash. You have 8 lanes on a flow cell, so you can sequence 8 different non-coded, non-barcoded samples. But if you have decided to add barcoded adapters to your library preparation, you actually could sequence, depending on what coverage you need, up to 96 samples in a single lane. So it all depends on how complex the sample that you're looking at, and also how much coverage you need for that sample. Let's see. So what I think is the sort of brilliance of the Illumina sequencing, and why I think it's really taken a hold on the next-gen sequencing arena, is the actual sequencing by synthesis. So in this case, you're using reversible terminators. So if you remember dideoxy sequencing in terms of Sanger sequencing, once you added the dideoxy, there was no more ability to extend the length of that fragment. In this case, they have developed a system where you will have your primer, and then you will incorporate all 4 nucleotides at the time. So remember, for the ion torrent, you can only flow 1 nucleotide over your chip at a time. In this case, you can add all 4 nucleotides, each one having a different fluorescent label. And wherever they find their complement in the different clusters, they will bind. Then you wash off, obviously the excess, and then you'll image, and you do a 4-color imaging. So wherever you have a C, you'll have a green dot and a yellow dot, and for the T, you'll have a red dot, for example. And then they have enzymes that will not only cleave the fluorescent tag off and wash it away, but they can regenerate the hydroxyl group, which is required for you to add the next nucleotide. And by doing that, you've now come back to the beginning without having to change anything about your flow cell and find out what the next nucleotide is. So you repeat it. You have 4 nucleotides. You wash off the excess. You color image. You get particular spots and positionally, right? So you take another picture, and what you can say then is that you'll have a green spot here because you added a C, and if you look at the next image in that same position, if you added a G, you'll have a yellow detection of the yellow fluorescence, okay? And you just repeat those cycles over and over again until you've reached the read length that you want. So this is what you will see. So each time you do a sequencing run, you will generate an image, and so it's positional and color-coded. And so you would go down, or hope the bioinformatics program on your system will look at that position in the flow cell. And if it sees green, blue, red, green, yellow, red, okay, then you can translate that into CAT, CGT. Okay? So each one of these dots represents the next nucleotide that was added to the cluster that you generated on your flow cell. And so, for example, here you see multiple green images in the same position, and you would read that as 6 Cs in a row. So the ability to run consistent sequencing reactions without having to, change out anything or be stuck by a dideoxy is what makes this so useful. And this is why the Illumina synthesis method can generate so much data. So, and because of the ease of that, and because of it takes about 3 to 4 days for the instrument to run, and the amount of data that it can generate, this is why it has been highly adapted in many of the clinical diagnostic labs. So those are sort of the background on the main next-gen sequencing platforms. And I wanted to put up one slide for the next generation sequencing, because this is coming down the pipe not very long. So PacBio is one of the instruments that's been around for a while. That's a long-read single molecule instrument. But recently we've heard in the news again about the Minion from Nanopore. And this is fascinating because the design of the instruments is small. One of our colleagues here is actually has the chance to do some experiments on the Minion, and it fits in the palm of your hand. And you are now sequencing a single molecule at a time. So in terms of sample prep, it's less complex. You get a much longer read length than you do with the traditional next-gen sequencing platforms. The different methods are sequencing by synthesis, or in the case where I'm showing you here is you pass a molecule through a nanopore, and it uses atomic force microscopy to do the sequence for you. The challenge right now is with any new technologies, the error rates are quite high. So in terms of its application to the clinic, it's certainly not there yet, but it's incredibly exciting because it has the opportunity to be a system that could be run in less developed countries, because you don't need a large instrument to transport. This is something you can pack in your suitcase. But just to keep your eyes open for these two technologies. I think, as anything, it's going to move much quicker than it did in the past, and I think as soon as it is developed to a point where the error rate is lower, it will have a great use in a lot of different molecular diagnostic labs. Okay, so I'm not going to really go over this aspect of it, but I did want to mention, it's great to generate the sequence, but you really need a solid bioinformatics pipeline afterwards, because you can generate a lot of data, but if you're not able to look at it and weed out the noise and validate what you find, then it's of no use to the patient. And so our students are actually quite lucky. They have in their senior year a whole year of bioinformatics. One of my colleagues, Dr. Shrivastava, actually teaches them bioinformatics as we go through the different techniques, and also goes through, in these last 3 weeks, first line analysis for next-gen sequencing. So this is very important from here on out, because you can generate as much data as you want, but if you can't collate it or and pull it into a story and get the information out that you need, then that information is useless. So again, in the bioinformatics analysis, you're looking for SNPs that are associated with clinical phenotypes. You often find novel SNPs. You have to talk about SNPs that you find that you don't know what the impact is, and then you still have to validate. So the ion torrent is taking over some of the validation process for some labs. They will run the sequencing on the Illumina, and they will validate on the ion torrent. But traditionally, still Sanger sequencing and amplicon sequencing is really the gold standard for validating any mutations that you might find in your next-gen sequencing. So you have a lot of uses for next-gen sequencing. I've listed them here. I'm just going to go over a little bit about whole exome sequencing. You know, because protein coding genes make up about 1% of the genome, but 85% of the mutations with large effects on diseases. This was the method of choice when sequencing an individual genome was still cost in the tens of thousands of dollars. So it wasn't practical to do a whole genome sequence for a patient. And so they developed whole exome sequencing to sort of harness the power of the next-gen sequencing platform for clinical use with patients. So this is just a schematic diagram. You generate your library, as I said before, and then traditionally or initially, they developed sort of a capture, slide capture, where you would generate probes for all of the exons and areas that you were interested in. And then you would flow your library over the top of that chip and grab all of the sequences that were homologous to the exons that you were interested in looking at. So you could do a small array up to a whole, a whole exome array. And those sequences that were from non-coding regions or regions that you weren't interested in would be washed away, and you would get a very targeted, very specific group of fragments to sequence. And so in this way, you could get rid of all the stuff that you didn't want and use the next-gen sequencer to get some very good data on just the specific regions that you were interested in. So originally, this was a slide-based protocol, and then, of course, they developed liquid, which is, in terms of automation, was a necessity because these were not automatable at all. And as soon as they could do it in liquid capture, then at least at the Genome Center, they put this whole process on a liquid handler and could automate it. And so you get a very targeted library, which you would use whatever platform you wanted to sequence and then generate your information. So what are the pluses of whole exome sequencing? Well, obviously, compared to the genome, it's a huge reduction in complexity. And if you're thinking about it in terms of offering a diagnostic assay for a patient in the clinic, it would be a significant reduction in sequencing costs as well, and also a reduction in time. So if you're up and running and you have a solid pipeline, you can generate thousands of exomes in the time it takes you to do whole genomes. And in the sense, we still don't know a lot about what the non-coding regions do or whether they have clinical significance. So in terms of healthcare, that would make a lot of sense just to stick with the coding regions. And the computing time needed to look at the data from your exomes is about 1/15 that for the whole genome, and it's probably less than that now. And again, it provides, most importantly, a focus on the regions where clinical molecular geneticists can give you the best interpretation of what they find, and hopefully it has some benefit for the patient. And you'll often see WES as the sweet spot in genomics, where you can get the most information for a moderate cost. So what are a few of the minuses? Well, again, it doesn't address any variants in non-coding regions or even non-coding exons. And I think we just don't know what we don't know at this point. I'm sure we will find there are non-coding regions which have significant effect on disease, and we won't know that until we find it. So in the meantime, we will be missing some things, and that may be why certain complex diseases are not 100% defined by the mutations that we find in exons. You can design your whole exome targets to include transcription sites, but typically it doesn't look at these transcriptional regulation sites, and it has had a limited ability to resolve copy number variants. It's not useful necessarily for looking at balanced structural alterations like inversions or translocations. You don't have a very good ability to pick up uniparental disomy, and again, with short reads, sometimes Fragile X and trinucleotide repeats, they're not able to be characterized. So it has its limitations, but in terms of offering information to a patient who is searching for a cause for something, it still, in my opinion, would outweigh the minuses. But again, it's often a cost-benefit issue. So we've all seen this slide a million times, but I think this is the most updated one that I can find. And what you can see now is, in terms of the clinic, the cost of doing a whole exome has dropped as low as $1,000. So now we're at the point where it used to be a benefit to do exomes only because of the cost, and now we're almost at the point, if not already there, where doing the whole genome is equally as cost effective in terms of just the price of doing it as it is of doing a whole exome. And so we've all been talking about the $1,000 genome for a while, and I would say we're pretty close to being there now. So, but there's a difference when you're looking at next-gen sequencing in terms of the clinic versus research. So in the clinic, you really need high coverage, and that means you need to cover that area that you're interested in as much as you can. I mean, 1000x is what you aim for because you want the low false positive rate, right? This is an individual whose potential therapy could be decided based on some information that you give them from your sequencing. So if you're in research and you're just sequencing or looking for changes, oftentimes your metrics are a 20 to 30x range. At the Genome Center, they tend to go a little higher than that, but in terms of research, it's much lower than what you would want in the clinic. And originally, you would just look at a panel of genes based on the individual's clinical symptoms. You know, you have much more freedom to look at a larger number of genes in research because you're obviously most likely searching for new changes or different mutations in different genes. And you really want to be able to detect in the clinic the full spectrum of the mutations, and it has to be certified and licensed in order to think about getting reimbursement from your insurance company. Whereas other than IRB approval, no accreditation is required for research. So it's a different kettle of fish once you head towards using this technology in the clinic. And what you're trying to do for the patient is, you know, compare their normal and their tumor DNA and be able to tell them something genetically that might give them an idea of their prognosis or might define the treatment that will most benefit them for their disease. So the pipeline itself probably doesn't look much different than if you're ordering traditional assays that doctors order for their patients all the time now. You know, you'd have a patient that would come in and talk to the physician, and they would order the next-gen sequencing. You'd take the sample, it would go off to the lab would sequence it, it would base call, it would do an alignment, it would call your variants, you would compare it based on your, the knowledge base that's out there, what information, if any, is known about that mutation and how it translates clinically or in an actionable way to the patient. You would generate a clinical report that would be entered into your electronic health record. That information would be sent back to your physician, and then they would have a discussion with the patient about what that information is telling them. So I think when a physician looks at ordering next-gen sequencing now, the first thing they probably have to consider is the clinical utility or the net health benefit, right? Are they really going to be able to tell the patient something useful that might help them deal with whatever clinical issue that they have. You know, one of the downsides is it has to be an individual evidence-based approach for each patient. So you have to think for each patient whether it's a beneficial thing to do an exome or a whole genome sequence. And one of the areas that's becoming tricky now is you have to be able to separate the information with medical utility for that which is unclear or which we really don't know functionally or clinically what that means for the patient. So this is an issue in Iceland now. So they've been sequencing their entire population for years. And so they're far enough out now that they have information on people which they think has potential actionable information about risk of developing certain cancers, but they're not really sure how to give that information to the patient. So the technology moved a lot faster than the ability to decide how to deliver that information, and also information on some things that they can't do anything for them. So this is a big issue for whole genome sequencing. You are going to find things if you go looking, and for most of those things we may not know whether it means anything for the patient or not. So you have to be able to sort of distinguish what's useful and what's not, and then based on that determine whether exome sequencing is useful for the patient. Most of the time, up until recently, the application of next-gen sequencing typically came after, you know, a patient or a family had exhausted all other testing options. You know, maybe it's a useful next test, right, leading to a clinical diagnosis or even treatment options. Oftentimes, the initial studies that were published had to do with a patient who had a clinical presentation that was unclear or atypical, and there are multiple genetic conditions and a differential diagnosis, so they would want as much information genetically to try to sort of figure that picture out for the patient. I'm going to spend maybe 2 slides on a particular project that I was involved in when I was at the Baylor Human Genome Sequencing Center. And actually, if you're in the Houston area, Baylor's actually using this in some of their commercials now for their hospital, this particular sequencing project. This was a pair of fraternal twins that had been, over the years, diagnosed with DRD, which is dopa-responsive dystonia, so it's a neurodegenerative disease. But at the time, they had been analyzed for the 2 main genes that were known to be involved in this syndrome, so the TH gene and the GCH1 gene, and I've just given you sort of the pathway for these neurotransmitters here. So if there were mutations here or here, then your neurotransmitter production was not correct, and you would have clinically heterogeneous movement disorders. So these twins were failing, and so their father actually worked for Life Technologies at the time, and so they offered to do their family sequencing using the high throughput sequencing to look at their genomes to see if they could actually identify any potential causative genetic variant. This is a typical case where they had spent, you know, a decade or so at a zillion different doctors trying to figure out exactly what it was that was ailing them. And even though they fit into the clinical diagnosis of DRD, they didn't seem to have the mutations that were known to be linked to that particular disease. And so at the Genome Center, they sequenced the 2 parents, the unaffected sibling, and then the twins. And what they ended up finding was that they were compound heterozygotes with different pathogenic mutations on each of their alleles. So the father was heterozygous for the mutation, the arginine to the glycine, and then the mother was heterozygous for the mutation in this area. And the twins had inherited one of the pathogenic mutations from the father and one from the mother on different alleles, which gave them the DRD. So it was interesting at the time because this gene had not been associated with DRD before. But if you go back, it is involved in the generation of these neurotransmitters, or actually here, it actually is involved in making a cofactor, which then is involved in making dopamine and serotonin. So this was a novel finding at the time, and what they did was they actually added 5-hydroxytryptophan, which is here, into their treatment. So they were on L-DOPA, and that was working, but it was failing. And so just by adding this, which was available over the counter, they were able to counteract the effects of the DRD by getting around the mutations here in the SPR gene. They were giving them the cofactor that they were actually missing that allowed them to make serotonin. And so this particular study has actually sort of changed, has directly influenced the clinical management of people with DRD. And it was fortuitous for this family that the fix for that mutation was something that was readily available to them. And these children are, I think, now in college, and the girl is actually running track, and they're doing very well. So this is one of the first real successful clinical applications to using next-gen sequencing to make a significant difference in an individual's health care. So just in the last few slides, I thought I might bring up some of the challenges for implementing next-gen sequencing in the clinic. I don't have answers for these things, but some of these are from reviews written by Elaine Martis, who's at the Genome Center at WashU. But as in most things, when technology moves fast, the system that's there to support it hasn't caught up. So in the existing paradigm, we're used to only looking at hotspot mutations, right? And only sequence from tumor tissue. So if you're doing next-gen sequencing, as I said before, you're going to detect all variants, somatic and germline. And in order to distinguish them, you should be sequencing some normal as well. But that then doubles the cost. So you have to decide, is it enough to sequence the tumor, or do you need some more information? Okay? And oftentimes, if you have novel mutations, they may not indicate pathogenicity, right? So you may not know whether it actually affects a gain or loss of function, and you may not have any information on how that would interact with a particular drug. Okay? One thing that Baylor Genome Center saw some years ago and is part of their mission is physician in training. So medical education is catching up now, but previously there was no real system for teaching physicians when it was appropriate to order next-gen sequencing, and then more than that, how to have the conversation with the patient about what the information was telling them. So they, along with other medical centers now, are developing user-friendly decision support tools to help the conversation between patients and their physicians, and make sure that the patients understand the information they're getting, and also to make the physicians confident in what they're ordering, and especially how to interpret those tests. And then there's really no straightforward mechanism to communicate the results of what each individual clinical lab is finding, because these are single patient studies mostly. And so you may find some interesting mutation in a gene related to a disease, but if you go through the traditional mechanism of writing the paper, having the paper reviewed, okay, that's a long time. And what really needs to be developed is sort of a real-time community-friendly database where you can share that information, because other labs around the country who are starting to do next-gen sequencing may come across a similar case. And if they have some place to go where they can see that it's been shown elsewhere, that would be much more effective in moving next-gen sequencing into a traditional diagnostic tool in a timely fashion that can be used in an actionably clinical way for the patient. And then something that we recognize here at MD Anderson is we have to train the workforce. And so it's been very important for us to bring next-gen sequencing in some form on board for our molecular genetic technology students, because they'll step in the lab after graduating and they'll see the ion torrent and they'll see the MiSeq, which is the smaller version of the Illumina, and at least they go in with an understanding of what that technology is and what kind of information you can get from it. Some other challenges, as with all labs that need certification, the reference material for validating the assays have not caught up with that. So people are now looking at developing multi-analyte calibrator materials. One is called a control plasmid spiked in genome. So you have a plasmid that has about 1,000 bases of sequence with a specific mutation near the middle of your insert with a 6 base pair molecular tag on it, and that's in with the sequencing reaction. And so since it's tagged with a barcode, it will be binned separately, and you can take a look at that. And so that can be a way of measuring the performance of your sequencing assay, and then also your ability to do the data analysis out the other end. This is definitely an area that needs to be developed, and of course, all the labs that are working in next-gen sequencing are developing their own, as well as sharing them among different labs. And then again, the importance of the bioinformatics. You've got to have the best coverage. You have to keep your error rates low. You've got to have informatic tools, not just that bioinformaticians can understand, but physicians, again, who are getting that information so they can really communicate to the patient, the significance of the information that they're getting. We're generating a lot of data, so storage is always going up. And then the ability to link it to the electronic medical records. That's actually very important as well. And just a few practical challenges. Since I spend a lot of time in the lab, I'm always thinking about the practical challenges. So the one is the tissue prep method. So formalin and paraffin embedding has been around for 100 years. That's the practice. And even though new preservatives are coming out, if you're going to have widespread use of next-gen sequencing, you can sequence from formalin-fixed, paraffin embedded, but it's not the greatest. So we need to move forward with finding different ways to preserve tissues, and that's going to be difficult to get those standard practices changed, just because they've been in practice for so many years. But if next-gen sequencing becomes a sort of standard assay and higher throughput, then clearly the type of preservatives that the tissue is stored with will have to change. And in terms of cancer, since we think about that a lot at MD Anderson, we have to consider tumor heterogeneity. So the bulk tumors, we all know now, consist of subpopulations of clones. So we have to think about, are we sequencing just one subpopulation based on the sample we've gotten, or can we distinguish the different subpopulations from our next-gen sequencing in some way, because those subpopulations may have substantial clinical implications. Most often you think about that in terms of metastasis, that one subpopulation of a tumor generates the metastatic disease down the road. And then the limited amount of tissue, we're getting better and better at seeing tumors, for example, earlier, which means they're smaller. And so the material you're getting from biopsies is smaller, which is a good thing. But then if your tissue becomes limiting, we have to make sure the next-gen sequencing can generate data in a low error rate with the limited material that we have. And so that's why I think maybe these third-generation sequencing platforms are really going to be the wave of the future as the amount of material that is accessible for these assays decreases. And then, if you're talking about a patient who's on a second-line therapy that requires a new biopsy, repeat sampling is neither standard of care or covered by insurance at this point. And then the way that we design clinical trials these days, especially Dr. Martis has talked a lot about how we need to change that and do more basket trials. So instead of looking at a gene in a particular disease or cancer, to do genes and look at multiple cancers, for example, because now we're seeing that some genes we thought were only specific for one disease can actually be involved in many. So I think, though, for me, one of the most exciting aspects is you know, if you're able to generate a person's whole genome quickly and effectively, and with the increase in immunotherapies, you know, down the road, we may be able to, in a very timely fashion, generate patient-specific vaccines based on the genomic makeup of a person. And that may be a significant advance in terms of treatment of cancer, for example. So I think the ability to get all that information has many applications, exciting applications for the future. I put this slide up because, you know, the commercialization of next-gen sequencing, it's here already. So Veritas Genetics is advertising their $999 whole genome with an app and access to a genetic counselor. And that's very exciting. And as George Church, who's, you know, been in sequencing for many years, says, now that the whole genome is accessible, it will replace all current genetic tests because it's all genetic tests and much, much more. What bothered me a bit about that was the next frame down was showing how, based on your genome, they could tell things about your athleticism and your youthful skin and things like that. So this, for me, I think is coming, but I don't believe we're there yet. There's a lot of aspects of next-gen sequencing. We can generate the data, but what's missing is, what is the data telling us, and where does it go from here? So, but just know that now, you know, you too can get your genome sequenced for $999. So, but for sure, whatever the challenges are for next-gen sequencing, it is and will continue to change healthcare. So thank you very much for spending time, and I hopefully can answer a few questions.
Okay, Dr. Newsham, thank you for a very informative presentation. We are running out of time, so if we may have enough time for one or two questions, if you do have them, please submit them by typing them into the text box. And so I'll wait a couple of seconds. If there are no questions, then I will then conclude with the conclusion remarks, and then that will be the end of the presentation. So right now, I'm looking at the chat box. I don't see any questions posed. In that case, thank you all for participating in this webinar. Shortly, the webinar participants will receive an email from AGT that includes a link to an evaluation. Please take a few minutes to access it and